Welcome back, dear students. In the previous class, we learnt about different kinds of glands that exist in the digestive system, like salivary glands, gastric glands, liver, pancreas, and intestinal glands. And we have learnt about the secretions of them as well. In this class, we are going to learn about the secretion of liver and the digestion that takes place in the intestine. So the most of the digestion takes place in the intestine, as I said earlier. Small intestine is also the site of absorption. Hence we learn about the process of absorption as well. As I said earlier, digestion of carbohydrates starts in the mouth with the action of salivary amylase, which is also called as ptyalin, and alpha amylase, and digestion of the proteins initiated in the stomach. We learned that in there HCl converted prorenin and pepsinogen into renin and the pepsin, and they are converted into dipeptidases. And the complete digestion of carbohydrates, proteins and the fats is done in the small intestine with the action of bile juice, intestinal juice and pancreatic juice. Let us learn what are the enzymes present and their action as well. Okay, let's go. Action of bile juice. Bile juice is produced by the hepatic cells of the liver and it is stored in the gallbladder. As the food enters into the duodenum, gallbladder gets the signal by the hormone called cholecystokinin and starts secreting this bile juice. Bile juice has no enzymes in it. Still, it helps in the digestion of fats by the process called emulsification. Bile juice has two components in it, bile pigments and bile salts. Bile pigments primarily give yellow color to the stool, which is the undigested food. Due to some malfunction, due to the attack of certain pathogens, these bile pigments called bilirubin and biliverdin releases into the bloodstream. So when it released in the bloodstream, the eyes of the patient and the skin converts into pale yellowish color. The disease is called as jaundice, and it is also called hepatitis. Mostly it is due to the viral attack, which is hepatitis virus. There is another substance called urochrome in the bile juice, which gives pale yellow color to the urine. Bile salt like sodium chloride and sodium deoxycholate helps in the process of emulsification of fats. Due to action of bile salts, fats converted into micelle form, which allows the lipases to act upon it. Here you are able to see a fat globule and which has hydrophobic ends, but due to the action of bile salts, it converted into micelle, where you can find two ends, first the hydrophobic end and the second hydrophilic end. Hydrophobic end does not have any affinity to the water, but hydrophilic end has affinity to the water, so that lipases easily act upon the hydrophilic end. So here you are able to see emulsified droplets where you can see the hydrophilic end is exposed. So through which the lipases acts upon the fat and converts them into glycerol. Okay, let's learn further. Along with the bile juice, pancreatic juice and intestinal juice is also released in the small intestine for the purpose of digestion. So as we learnt in the earlier, pancreas has two parts in it, exocrine part and endocrine part. This exocrine part, which is also called pancreatic acini, starts secreting this bile pancreatic juice. Pancreatic juice contains several enzymes in it and plays a very crucial role in the digestion of all the three food molecules such as carbohydrates, proteins, fats, and also nucleases. Let's see what are the enzymes present in them. Pancreatic amylase, which acts upon the carbohydrates, carboxypeptidases, trypsinogen, chymotrypsinogen, all of these three acts upon the proteins, and nucleases, which acts upon the nucleic acids, and pancreatic lipase, which acts upon the emulsified fats. Okay, there is another secretion called intestinal juice, which is also called as succus entericus. This acts only in the alkaline medium, which is secreted by intestinal glands, and pancreatic juice has the pH of 7.8, which is alkaline in nature. Succus entericus contain disaccharidases and peptidases. Disaccharidases acts upon the disaccharides of the carbohydrates. Peptidases acts upon the dipeptides and the tripeptides. Okay, let's learn further, and each enzyme action and how it digests the different food molecules is learnt in the next slide. Let's see. Let's learn about the digestion of carbohydrates in the small intestine. As we studied earlier, digestion of carbohydrates starts in the mouth, where the alpha amylase or ptyalin acts upon the carbohydrates and they convert it into dextrine and maltose. We have learnt about it in the earlier. And in the small intestine, the pancreatic juice contains pancreatic amylase, which converts the carbohydrates into maltose, sucrose, and lactose. All these three are the disaccharides. Okay, so carbohydrate is a polysaccharide which is broken down and converted into maltose, sucrose, and lactose with the help of this pancreatic amylase. And succus entericus contain disaccharidases like maltose, maltase, sucrase, and lactase. These three are the enzymes which are also called as disaccharidases. Okay, so these disaccharidases helps in the digestion of the disaccharides and convert them into the monosaccharides. Let's see, maltose is converted into glucose and another component of glucose with the help of an enzyme called maltase, which is a disaccharidase. Okay, and the next, sucrose converted into glucose and the fructose by the action of sucrase, which is also known as invertase. Okay, and the lactose convert into glucose and galactose by the action of lactase. All these three, maltase, sucrase, and lactase, are called disaccharidases which exist in the succus entericus. Okay, this is how the digestion of carbohydrates take place. It starts in the mouth and the completed in the small intestine. So glucose is a simple soluble form of the carbohydrates which enters into the bloodstream by the absorption. Okay, let's see further. Let's learn about the digestion of proteins. We all know that the digestion of proteins is initiated in the stomach. So here I have given the pictorial view of what are the changes take place in the stomach. Let's see, there are inactive enzymes such as prorenin and pepsinogen converts into renin and pepsin, which are active, and this activation is because of the action of HCl. Okay, HCl converts this inactive form into active form so that they can act upon the proteins. Renin converts the casein protein called creatine present in the milk, converts them into calcium paracaseinate, and calcium paracaseinate are further converted into peptones by the action of pepsin. Okay, casein is found in the milk, so the renin enzyme is mostly found in the infants, because the main source of food in the infants is the milk. Okay, and pepsin converts the proteins into proteases and peptones. This is the partial digestion of the proteins which take place in the stomach. Let's see what happens in the intestine. As we learned just now, pancreatic juice contain the inactive enzymes called trypsinogen and chymotrypsinogen. These are inactive and they convert into their active forms, which are trypsin and chymotrypsin, with the help of enterokinase and trypsin. Okay, so enterokinase activates and the trypsinogen into trypsin, and this trypsin enzyme has the capacity to activate its own inactive form. Hence, this process is called autocatalysis. Only active enzymes can act upon the food molecule. Let's see, proteins, proteases and peptones converted into tripeptides and dipeptides with the help of the enzymes called chymotrypsin, trypsin, carboxypeptidases and aminopeptidases. And tripeptides and dipeptides convert into dipeptides and amino acids, dipeptides into the amino acids with the action of tripeptidases and dipeptidases. These tripeptidases and dipeptidases are present in the succus entericus. Okay, this is about the digestion of proteins. The simple soluble form and absorbable form of the proteins is the amino acids. Okay, let's learn about the digestion of fats. Well, the digestion of fats is started in the small intestine and ends in the small intestine itself. We learned earlier that the fats are converted into emulsified fats with the help of bile salts. Okay, fats cannot be acted by the lipases directly. Hence, first they convert into the emulsified fats in the form of myceli. Just now we learnt about it. Emulsified fats converted into diglycerides and monoglycerides by the action of lipases. Okay, so pancreatic lipase converts the emulsified fats into diglycerides and monoglycerides. Monoglycerides are further broken down and they convert into fatty acids and the glycerol. These fatty acids and glycerols are the absorbable form of the fats. Okay, and nucleic acids such as DNA and RNA are digested by the nucleases. Nucleases are the enzymes. There are two types of nucleases. They are DNAase and RNAase. Respectively, they digest the DNA and the RNA. They convert them into nucleotides. Nucleotides has two parts in it, nucleoside and phosphates, as we learnt in the structure of DNA. So these nucleotides are acted by the nucleotidases, which is the enzyme, and converts into nucleoside and phosphates. Nucleosides further divided into sugar and bases by the action of nucleosidases. Okay, so this is the digestion about the nucleic acids. Let's learn further. Absorption of nutrients. So far we learned that carbohydrates are broken down and they converted into glucose. proteins are converted into amino acids, and fats are converted into fatty acids and glycerides. So all these are the simple soluble substances which has to be absorbed and enter the bloodstream. So the transport of food, simple soluble food substances into the bloodstream is called as absorption. Okay? So absorption is done at the villi, which is a finger-like projections that we find in the small intestine. They are actually non-ciliated columnar epithelial cells. So it is a layer of columnar epithelial cells which are non-ciliated, but though they are non-ciliated, they appears to be brush border surface because they contain microvilli on the villus. We studied about the structure of villi where we found this microvilli. Food molecules first they transport to the intestinal mucosa and then they enter the bloodstream. So this transport is by different means. They are active transport, passive transport and facilitated transport. Let's learn what are these transport mechanisms. In the active transport, the substances are transported from lower concentration to the higher concentration. So the movement of molecules from the medium of lower concentration to the medium of higher concentration is called active transport, which requires the energy supplement called ATP. Since they require the ATP for the transport, they are called as active transport. Okay, the next is passive transport, where we does not need any energy. In this type of transport, the food molecules transported from higher concentration to the lower concentration. We all know that in the diffusion is also take place from the higher movement of molecules from the higher concentration to the lower concentration. It does not require any energy because there is a gradient which is present. Next, facilitated transport, which means the transport is facilitated or helped by the proteins. Okay, so if the proteins are involved and they help in the transport of the molecules from one end to the other end, this kind of transport is called facilitated transport. Let's see some bit bank pH of the different substances of the digestive system. Saliva has 6.8 pH, which is slightly alkaline and slightly acidic, and HCl has 1.8 and 1.8 to 1.9 pH, which is a strong acid. Pancreas has 8.4 pH, which is alkaline in nature, and intestinal juice also has 7.5 to 8, which is a alkaline again. Okay, let's learn further. Now let us learn how these food substances gets absorbed. So glucose, fructose and amino acids are transferred to intestinal mucosa layer by the process of active transport, which require the ATP. From intestinal mucosa, they enter the bloodstream by the means of facilitated diffusion, which is done by the proteins. Fatty acids, glycerols converted into chylomicrons. The group of chylomicrons is called as micelles. Micelles transported to columnar cells by the facilitated diffusion, facilitated by certain surface proteins. Finally, micelles divided into chylomicrons. These chylomicrons goes into submucosa by exocytosis. Okay, so chylomicrons finally enter into the lacteal, which is also called as lymph vessel. Okay, so the fats in the form of chylomicrons enter into the lacteals. So Let's see what is the difference between chylomicrons and myceli. The main difference is the structure, is that myceli are just made of phospholipids, whereas chylomicrons are made up of triglycerides and phospholipids. Okay, so uh, glucose, fructose, amino acids are transferred to the mucosa layer by secondary active transport. From there they enter the bloodstream by the facilitated diffusion. Fatty acids directly cannot transfer as they are, so they convert into chylomicrons and they enter the lacteals, which are the lymph vessels. Let's learn. See the pictorial view of how the substances are absorbed. So first of all, this is the gut of the small intestine, and from the lumen of the gut they enter into the mucosa of the small intestine, the intestinal layer, and from there they enter into the small intestine. So between the gut and the bloodstream, the food molecules have to enter into the intestinal layer. Okay, let's learn further. Let's learn some technical terms. Absorption means the absorption of food substances with the help of villi, where the food molecules transferred from the lumen of intestine to the bloodstream. Okay, and assimilation means whatever the food substances enter the bloodstream, they goes to the cellular level and transferred and transported to the each cell. So this passage of the food molecules to the cellular level is called as assimilation, and after reaching to the cellular level they undergo the cellular respiration process, and they start releasing the energy in the form of ATP. Digestion means it is nothing but the elimination of the waste, undigested waste. It is also called as defecation. Role of large intestine, let us see here. Water is reabsorbed from the undigested waste. So the most of the water is digested in the small intestine itself, and whatever the remaining water is there is absorbed by the large intestine. Mucus is added for the smooth passage of fecal matter. Okay, the feces is stored in the rectum and eventually expelled from the anus. This is done by the peristaltic movement. Actually, peristaltic movement uh, exists right from mouth to the anus. Okay, so it helps in the passage of the food. Let's see some gastrointestinal hormones which helps to stimulate certain glands to release these hormones, or intestinal juice, pancreatic juice, bile juice, or whatever it is, they help in the digestion. So the gastrin is released by the epithelium of the stomach, and it stimulates the release of gastric juice from these walls of the stomach. Enterogastrin is released by the epithelial layer of the duodenum, and it inhibits the gastric activity. Okay, and the secretin is released by epithelial layer of the duodenum, and it acts on the pancreatic acini to release or to secrete pancreatic juice. Cholecystokinin, it is also called as pancreozymin. It is again released by the epithelial layer of the duodenum, and it acts upon the gallbladder to secrete the bile juice. Okay, cholecystokinin stimulate the gallbladder to release bile juice. Enterokinin, it is again released by the duodenum, and it stimulates the secretion of succus entericus from the intestinal walls. Villikinin is another hormone which stimulates the villi to absorb the food molecules at a faster rate. Let us learn some of the diseases where the digestive system gets affected. Jaundice is one of the disease where bile pigments, bilirubin and biliverdin, released into the bloodstream, due to which the skin and eyeballs appear in a yellowish in color. This is due to the hepatitis virus, and hence disease is called hepatitis and jaundice. Vomiting is due to the reflex action which is initiated by the vomiting center in medulla oblongata of the brain. Diarrhea is nothing but the loose motions. Due to this, the absorption percentage gets decreased and the food molecules are not absorbed, and heavy loss of water also occurs, which leads to the dehydration. Constipation is nothing but uh, the the retaining of the undigested waste more than 2 days in the rectum. Indigestion is very common problem these days. It is due to the intake of spicy food, taking over food, and due to depression and anxiety. Let's see some of the calorific values of the different food substances. 1 gram of carbohydrate and 1 gram of protein, they yield 4 kilocalories of energy respectively, and 1 gram of fat can yield 9.0 kilocalories of energy. Dear students, this is about the first chapter, digestive system. Hope we will learn further units. Okay, thank you.